Afternoon, sir.
Good afternoon. Good afternoon.
Congratulations, sir.
Thank you.
Okay, now you are in Belgaum, na sir?
Yeah. Now I'm back in Belgaum.
Okay.
A very good afternoon to all. Now we are in second session of AICT sponsored FDP on Nutrigenomics Unveiled Frontier in Healthcare organized by Bharati Vidyapeeth College of Pharmacy, Kolhapur. Now I would like to request Ms. Jitnesa ma'am to introduce our guest speaker Dr. Subarna Roy sir. Over to you ma'am.
Thank you. It is my immense pleasure to introduce Dr. Subarna Roy sir. Sir is presently working as Director, ICMR NRTM, Department of Health Research, Ministry of Health and Family Welfare, Government of India, Belgavi. Sir has worked in various ICMR institutes since 1998 at Kolkata and Port Blair before joining NIE Chennai in 2020. He has also worked with state administration and district health surveillance units across Karnataka for identification of infectious disease outbreaks, monitoring of drug resistance patterns, and helping in control and prevention, and in committees in setting up of viral diagnostic labs in various districts of Karnataka. His area of expertise include microbiology and molecular epidemiology of infectious diseases. Sir has undertaken several molecular genetic studies on authentication of medicinal plants, documentation and preservation of germ plasms and genetic fingerprinting of medicinal plants of Western Ghats. He has established microbiology, molecular biology and biomedical informatics labs at NITM Belgaum. Sir is research guide for PhD students of Pondicherry Central University, Shivaji University Kolhapur, KLE University Belgaum and Symbiosis International University Pune. 8 research scholars have been awarded with PhD while 4 are pursuing PhD under his guidance. Sir has more than 97 publications and has filed 3 patents. Sir has been a part of 26 extramural projects as a principal investigator and co-investigator worth rupees 32.18 crores. He is member of Research Advisory Committee of Multidisciplinary Research Units of KIMS Hubli. He has served as member of Academic Council of KLE University Belgaum, Chairman of Ethics Committees of KLE University Belgaum and Government Medical College KIMS Hubli. Sir is reviewer for more than 50 international journals, including the American Journal of Tropical Medicine and Hygiene, WHO Bulletin, and many more. Sir was a part of a scientific mission to NIID Tokyo in August 2016 to develop scientific and strategic Indo-Japan collaboration in antimicrobial resistance. Sir is recipient of many prestigious awards like Medical Innovation Fund Award by ICMR in the year 2007, Dr. S.S. Mishra Memorial Award by National Academy of Medical Sciences in the year 2008, ICMR Major General Sahib Singh Sokhi Award in the year 2009 by Honorable Minister of Health and Family Welfare, Government of India, 2013. With this brief introduction, I request you, sir, to begin with the session. Over to you, sir.
Thank you. I hope I'm audible.
Yes, sir, you are audible, sir.
Okay, thank you. So I will share my screen. Is my screen visible?
No, sir, still not.
I hope it is visible now, I think.
Yes, sir. Yes.
Yeah. Okay. So a very good afternoon to all of you. And at the onset, I would like to thank the organizers, Dr. Tambole and the colleagues at the university to have called upon me for this particular lecture on this validatory function of this program. And we are also celebrating the India International Science Festival. So it is very apt that we are conducting this session during this time. And it is always a privilege to talk to you in this forum. And I think I had spoken in this forum a couple of years earlier also. And again, I would like to thank the organizers for having me here. And as was told, actually, I was in Belgaum for quite a long time, for almost 12 years. And then I was in the National Institute of Epidemiology for the past 1 year. And now I have come back. So I've joined back this week itself on Monday. So what I will do is that I will talk about molecular tools in pharmacogenomics. I well understand it is more of a nutrition kind of a thing, so more of nutritional aspects. But I have very limited knowledge in nutrition. But what I will do is that I will go through some of the techniques that are available in which we are actually slowly turning back to personalized medicine. And our modern tools have actually helped in this process. And that is what I'm going to take you through. Basically, I will start with the holistic and personalized nature of ancient medicine and the evolution of modern medicine. Then the technological advancements and modern tools that we have with PCR, sequencing, microarray, and their applications in personalized medicine. So the medicines actually started with evidence-based. Basically, there used to be diseases and conditions which used to be observed, and then the problems or the ailments were diagnosed, and treatment was initiated, and response was monitored, which was again adjusted depending on the outcome. And then again, if there was not much of an improvement or changes required, so they were again diagnosed and the cycle would repeat. So that is how evidence-based medicine started. And as one of the oldest scientific tests had said that superior doctors prevent the disease, mediocre doctors treat the disease before it is evident, whereas inferior doctors treat the full-blown disease. So basically, it is prevention and then treatment before a disease becomes evident. These form the priority for treatments. Now, in the beginning of modern medicine, that was also quite simple. And it started with three fundamental questions that need to be asked. What is wrong? The diagnosis part of it. What is going to happen? The prognosis part of it. And what can be done about it? So the treatment of it. So it was very simple that way. And the practice of this medicine was also very simple. It used to be certain home remedies that we would be using. Diet was a major issue, and I'm sure you might have had a lot of discussions on this, particularly with nutrition. Faith was a major factor as far as healing was concerned. Even sweet words and assurances sometimes would help people. The very fact the doctor says that don't worry and you will get well soon within 3-4 days, that itself would actually make people much healthier and faster in recovery. Then slowly the horizons of medicine started widening. With the march of time, the questions also got more and more in number. Answers also got more and more complex. Technology started replacing even sometimes common sense and even bedside wisdom. And the personal role of the physician as a friend, philosopher, and guide has now become more and more nebulous. So what are the more questions that needed to be asked? When did it happen? The etiology, how did it happen? The pathophysiology, what occurred inside? Metabolism, biochemistry, neurochemistry, endocrinology. What is the root cause? The environmental effects, the genetic factors. How do we confirm valid conclusions of what we have learned? Similar to kind of evidence-based medicine that we talked about. The research methodology component comes here. Technological advances further complicated the impact of this evidence-based medicine. Science-based medical education, the CME program that we call, actually originated in USA in 1910, and that raised the standard of medical education, promoted as one advancement of science. You are all pharmacists, again, this is part of your equivalent of CME. So with the development of technologies, we actually get our knowledge also levels improved. The specialties also mushroomed and medical education of high quality progressed. Medicine became compartmentalized. Growth of specialization impacted on the practice as well as on society. There are several issues with ethical and legal framework emerged. Population growth, urbanization, globalization also impacted medicine. The progress in pharmacology and unprecedented growth in the drug industry had also its consequences, and you know much better in this regard than anybody else. Lifestyle changes, it had its own impact on health and society. There was increased impact of health consciousness and health education. Internet and web-based education actually became aids and also challenged medical profession. Economics and cost now dictate medical practice. And as we had growth of sub and super specialties, you can just see this slide, how many number of disciplines and sub-disciplines have we come up with? There are more. Can you just imagine the kind of complexity that has come from a simple evidence-based medicine that used to be practiced centuries ago? Now, these have actually had much more impact because of certain technological advancements that we had. And particularly for this, three major advancement in technologies are responsible. The first and foremost was the discovery of the invention rather of PCR, the polymerase chain reaction, which happened during the 1985 to 87. Kary Mullis, as you know, was the inventor of that. Subsequently, the DNA sequencing was also a major tool that came to be used again in the decade from 1977 to 1987. At the same time, there was development of microarrays. So these three techniques actually changed how we looked at diseases. And that led to the advent of genomic medicine. This is how a polymerase chain reaction thermocycler looks like. And this is one of Biorad, but there are several other brands which are also being used. And nowadays it is a household word because of COVID-19 that, as you all know, PCR is the gold standard test for testing of COVID-19. So PCR has got a lot of applications. It has applications in diagnosis, it has application in identification, it has in characterization of different strains and subspecies. Other downstream applications for DNA sequencing, detection of mutations, predetermining sex of embryo, forensic sciences, linkage analysis, studying molecular evolution, ascertaining recombinant clones. So these are kind of applications that PCR has. So the basic principle of PCR is it is a molecular technique which is very, very sensitive. It is extremely sensitive. and it can actually able to pick up or identify and amplify even a small fragment of signal DNA theoretically into 1 billion times. And it actually imitates or replicates the DNA replication which happens inside the cell. When a cell is dividing, the cell is growing, so the DNA replication occurs inside our body. So it actually imitates the same process artificially inside a small tube, which happens in that thermal cycler, which I just shown you in the picture. It uses certain components or reagents, which we call as the components of PCR. One of them, major one, is the DNA polymerase. That is the principal enzyme that actually helps in actually magnifying or making copies of the template DNA, and such as DNA template for synthesis of a complementary strand from the raw materials, which are deoxyribonucleotide triphosphates, or in short we call them as DNTPs, in a replication process within the tube. And this is aided by 2 primers which are added, and this primer is designed by us as the starting material. These primers are specific to the region that we want to amplify. Say, for example, in SARS-CoV-2, in this present pandemic of COVID-19. So the type of gene that we would like to detect, we generally take a primer that targets that particular gene, and then we put it inside those tubes. So if that particular gene is present, then that primer will be extended through this DNA DNTPs and by use of this DNA polymerase, and we will get a positive amplification. If the template or the pathogen is not there, then it will not amplify and it will be a negative one. Thus, the starting point of DNA synthesis can be specified by supplying an oligonucleotide. In this case, we call the primer, that anneals or attaches to the template that is provided in the reaction mixture. There are several forms of PCR that are used for various purposes. We generally call it simplex, uniplex, or singleplex PCR when there is only one single target that we generally amplify. We use a single pair of primers for that. If we are targeting multiple genes or multiple regions or multiple targets in one single run, then we can call it a multiplex PCR, where we use 2 pairs of primers or more. AP-PCR, or arbitrarily primed PCR, or a random amplified polymorphic DNA PCR, often called RAPD-PCR, is used when we don't use a pair of primer but use only a single primer. We do not define the ends. That means we give only the starting point, but we don't define the end point of the sequence that we want to amplify. What it does, it generates arbitrary fragments on various places where it might arbitrarily go and bind in that template which is present in that reaction mixture. So it will land up in creating a multiple number of bands, which will appear as what we often call as fingerprints. So these fingerprints are generally used to match one another. So for example, a particular strain of organism that I isolated here, say in Belgaum, and you have isolated the same species in Kolhapur, and we do a RAPD PCR or a IP PCR, and then we compare these fingerprint patterns. If the comparison shows the same pattern, then we can say that these are most likely to be the same strain or same subspecies or the same substrain which is there in Kolhapur and in Belgaum. So this is the application of that. Real-time PCR is when we do the amplification when it can be seen as when the amplification occurs. This is what is presently done in COVID-19 testing. Normally, normally a PCR when we do, we generally have the amplification product, and then we do a electrophoresis, or we run it on an agarose gel to see whether amplification has occurred or not. Basically, it takes time. So, and your results will be either the amplification band will be visible, present or not. But in real-time PCR, you can actually see in your computer screen whether amplification is happening or not in real time. So say within a few cycles, if you don't see an amplification, then you can straight away say it is negative. You don't have to wait for the amplification to complete and then run the agarose gel. So that is the advantage. It is fast, And second advantage is that you can actually quantify the amount of DNA or RNA which was present in your original sample. So it is a quantitative test also. So you can actually quantify the amount of template that was there. So that can help in actually understanding viral loads and bacterial loads inside the patient serum if you are using that. There are various other forms of PCR, sometimes asymmetrical PCR, when we take two primers or a primer pair, but one of them is reduced 100-fold in concentration, so that the other strand actually increases many times. So that is why it is called asymmetrical. So we are amplifying both strands here, but one of them preferentially over the other. Inverted PCR is once when you choose primers in such a way that it does not amplify the portion in between these two primers, but the portion which is outside them. So that means its directions are, instead of facing each other, it is facing the opposite side. Hello?
Sir, slides are not moving.
Okay, when was it stuck? Which slide?
Still it is on first slide.
It is still on first slide.
Yes, sir.
Oh my God. Just a minute. Is it visible now, slides?
Yes, sir. Yes.
Yes, sir.
Now, number 2 slide. Slide number 2.
Now number 3.
Yes, evidence-based medicine.
Okay, so what I will do, I think the slide view is having problem. So I will show like this. That will be better.
Yes, sir. Yes, no problem.
So I went through all this. So these are what I had gone through.
Yes, sir. No problem, sir.
Yeah, so I'm just running them through. I will come back to that same portion where I was in.
Okay.
This is the PCR equipment that I talked about. Many of you must have seen it because now we have PCR labs across the country. These are the applications that I was talking about: diagnosis, identification, characterization, downstream applications of PCR. This is the basic principles of PCR that I talked about. These are the forms of PCR that I just explained: single plex, multiplex, APPCR or RPD-PCR, real-time PCR, asymmetrical PCR, inverted PCR. Then you have nested PCR. Nested PCR is something when you do one PCR, amplify one product, and then again amplify a certain subproduct within that product. So that very often happens in case of hepatitis B. So for certain viruses, we generally use this technique. And then there is a reverse transcriptase PCR. Reverse transcripted PCR, also called RT-PCR, is when you actually start with a RNA template. When the RNA is converted into a DNA and then the PCR is done, then it is called a reverse transcripted PCR. In case of COVID-19, it is both reverse transcriptase PCR as well as real-time PCR, which happens together. So here the RT stands for reverse transcriptase as well as real-time. So this is a real-time reverse transcriptase PCR that we generally do for detection of COVID-19. Now there are certain limitations for PCR. So as I said, that PCR technique is such sensitive that even one strand of PCR, one strand of DNA or RNA present can be amplified and it will show as a positive. So sometimes what happens that we land up in contaminations, which can result in false positivity. You may have seen in many of your friends' cases or whatever that sometimes they turn out to be positive, even though they don't have any symptoms or very unlikely to be positive. It has happened even with my wife once. She was found positive. I did second time, next day she was negative, and none of us had any symptoms. 5 of us were together. So this happens sometimes because of what we call as carryover. Maybe a sample which perhaps in the sample previous to my sample, say if there is somebody forgetting to change the tip or something during pipetting, so that can carry over to the next tube, and sometimes you may land up in getting false positivity. So these are kind of things that happen. So we have to be very careful in using PCR for this particular reason. So that is why we generally use single use of reagents. We use different rooms for DNA or RNA preparation, amplification, and post-PCR detection. The use of UV or gamma radiation to inactivate or to remove the contaminant DNA also is done. And clean work habit ensures both trouble-free and reliable results. Then comes the second important thing, which is the, the sequencing of DNA and protein sequences. I hope the slides are visible.
Yes, sir.
Can anyone confirm? Yeah.
Yes, sir.
Okay. So the DNA sequencing was introduced in 1972, and the technology has been evolving. The first sequencing was like we used to do Sanger sequencing, and now we have next-gen sequencing. For example, now the SARS-CoV-2 sequencing is done mostly in next-gen platforms, which were very limited earlier, but now it has been increased, and they are extremely fast, and they can be, we are sequencing whole genomes of viruses so quickly. And 10 years back, it would take months together, if not years, to sequence. So protein sequencing also started in 1970s, and you know the contribution of Hargobind Khurana and his colleagues in protein sequencing, and then the mass spectrometry and Edman degradation. These are again techniques which actually came in together. And in sequencing, the target regions are amplified by PCR, and then this DNA or complementary DNA, in case of RNA starting material, is made in vitro in laboratory. Then the sequences of these bases are determined, and then the translation product is cataloged. So we have both the DNA, the DNA codes, as well as the sequences of the proteins, and then we identify which mutation has caused which change in the amino acid sequences of these proteins. Those of you who are aware with the again with the COVID-19, you have the South African variant which has come up and which has been named yesterday. This again, these are all mutations which has happened in the sequence of the RNA of the virus, and that has resulted in, or likely to result in, changes in the amino acid sequences of the proteins that they are making. So this way they can be sometimes immune escape, they can escape even vaccines. So that is the worry, and that is what are the tools that are being used actually to identify these scenarios. So this is the progress in genotyping technology from 2001 to 2005. Now, even there are much of progress that has been made. So it started with ABI, TACMAN, and then we had Illumina platforms, and then now we have the next gen platform. The technology is completely different. So technology has been evolving, and the same thing is with microarray, where we had the advent of biochips. In something like these slides, you have various wells of minute volumes, where you actually do a kind of probe detection. So you have sequences, various sequences of your starting material, the DNA or RNA, which is complementary products, is put inside. And then you have the fluorescent level probes, of various probes, thousands of probes you can use in each of these wells to detect the presence of those particular amplicons or not. So you can actually literally see hundreds and thousands of genes, whether they are present in that your sample or not. So that is how microarrays are used. So it evolved in the early 90s, and two primary concepts which led to the development and use of this DNA microarray are the amount of a particular substance in a mixture of substances can be determined through probe or target interaction. So it is also a semi-quantitative method in which your starting material, the quantity also you can actually determine through the degree of fluorescence that you obtain, and the DNA can be labeled for detection as and when using the Southern blot technique. So in microarray, the principle is that it uses either the complementary DNA or the oligonucleotides as probes, and these probes are allowed to hybridize with fluorescently labeled target DNA strands, just like in Southern blot. And when they fluoresce, this fluorescence is detected either with a scanning confocal microscope or with a CCD camera. Then the data analysis is done, and then we determine the relative amounts of expression or presence or absence, as the case may be. And then the microspotting techniques, which is around 300 micron diameter spots. These are advanced techniques that can use even 10 micron, which can be placed in extremely high densities on a single slide. So that is how we improve the throughput and then the number. So that way the large scale applications are possible. The DNA microarray can test for tens and thousands of gene transcripts at the same time and conditions. It is a high throughput and it's ideal for three main purposes. First is to identify patterns. That means testing globally for disease specific patterns such as gene expression or certain gene signatures, often of a fewer than 50 genes can be characterized. It can measure patterns, say with the use of a certain drug or with the disuse of a certain drug, what is the pattern change that is happening. So the impact of the drug or the quantity of or the measured change can be actually quantified in this particular application. Only specifically selected and validated probes are used to test for these patterns. As pricing and ease of use improve, these focused arrays may eventually become regularly used in diagnostics and drug discovery. Even parallel processing is there, which is beyond the scope of this presentation, but it can be used whenever there is a process that will benefit massively from parallel processing. So basically it is multiple things which are done together. A geneticist used to study only one or few genes at a time earlier. Now we are studying several genes and thousands of genes together, and even assays which are actually looking at multiple organisms. Even say, hello.
Yeah. Hello.
Any question?
No, sir.
Any question?
No, sir. The voice is coming from the participant side. I will.
Okay.
Please continue.
Okay. I will continue. Yeah. So geneticists used to study mostly one gene at a time, but now what I want to say is that technology has allowed us to study thousands and thousands of genes together and then to compare them parallelly in one single experiments. The utility of microarrays is to give a big picture biology. The big data biology is actually coming in here, where what all the components and what all the processes are taking together in a particular cell. So we are not only understanding one particular reaction, we are trying to gather information about several reactions occurring together due to a certain interaction of a particular drug or a compound. The systems biology picture is coming here. Now, how do these components react and how they are processed, how they interact with the various molecules to sustain life. So this is one approach which happens to the entire cell when one particular gene process or a process is perturbed. So this kind of information we are able to generate together. So this is a pictorial depiction of what I just talked about. So we start with the DNA and its RNA product, or if it is a virus, then you have the RNA as the starting material, and then the proteins that actually are coded by this and the protein products. It can be sugars, it can be nucleotides, it can be amino acids, it can be lipidome, the lipids, and then When we talk about the study of these genes, then it becomes the genome. When we talk about the RNA product, it is transcriptome. When we talk about proteins, it is proteome. And all of these products taken together become the metabolome. So, and this actually determines the phenotype or the function of the organism or the pathogen. So, once we build the chips for microarray, the massive chips are massive PCRs are done, and this purification and preparation of the products are done. They are printed on slides, and then they are processed, and a hybridizing chip is made on one hand, whereas on the left hand side you can see the template preparation where you have the RNA or the DNA, and then the culture is made, and then they are harvested. The RNA is isolated, a cDNA is produced, and then they are labeled with probes, and then the hybridization happens with the printed hybridization chip. And then once we get the detection of the fluorescence in the CCD cameras or in confocal microscope, as I said, then this data is analyzed in computers to give you the reactions. The same thing, again, a description in a different infographic you have with the mRNA, the cDNA, the products, and then you take it into the chip, and then you detect the fluorescence. Basically, the same thing again shown with another cartoon and with different pictures. This is the arrays lab, particularly, which you can see the array lab. And this is how the biological question. This is the flow chart of the microarray tests, where you have the biological question, you have differentially expressed genes, the sample class prediction. Then on that we design the experiment, and then the microarray is done, the image analysis is done, and normalization is done. Then again testing, estimation, clustering, discrimination depending on your query or the kind of objective that you have in your study, and then you do the biological verification and interpretation, and you get back. So this is the workflow of this. This is a typical yeast genome expression slide, which shows the various levels of expression of the various genes. Each individual well is actually a particular gene which is being expressed differentially. So this analysis is done over the computers, and then the logical conclusions are done. Now these are used in the genomic medicine. Again, I come back to the practice of medicine. So these techniques are used in the Human Genome Project, which completed in 2003. It took a very, very long time to complete one genome, but now it takes only a few days to complete each genome. And with the advent of modern technologies and with the fast sequencing that we have in the next generation, in effect, the genome became a tool for enabling physicians to understand the biological and the disease variability in the patients. The concept of personalized medicine again came back. Understanding the variability between individuals allow for more targeted healthcare based on genetic differences. These genetic tests are powerful because they are able to diagnose the disease more precisely. They allow for selection of specific medication to best treat the disease, to predict the risk of disease before the symptoms occur, and that risk can differ from me to you or to somebody else, and manage the disease more effectively. So what are these genetic tests? This genetic analysis of the DNA of the individual. The amplification of the DNA is done at one or several regions by PCR, depending on what are the kind of information that you want to collect. And the sequencing and the data analysis or microarray analysis that I just talked about, or you can do a transcriptome or metabolome analysis. Many of you may have seen sometimes papers on the whole gut microbiome analysis, sometimes whole lavage analysis of the bronchioles. So these are kind of things that are now coming in. And because we know more specific information today, we can diagnose diseases more precisely and therefore provide more effective treatment. We can select specific treatment that best fits that particular disease and therefore target the medication to that particular disorder, avoid adverse drug reactions, avoid delay in false starts. We can also predict the risk before even the symptoms occur and therefore provide early treatment and take preventive action. You can remember the first slide that I talked about, that the best physicians actually are those who can actually prevent the disease. So this is where it is coming back to. And then manage the disease more effectively by eliminating unnecessary treatment, by providing better timing, and by adjusting the treatment as the disease changes. Again, the evidence-based medicine slide that initially you missed, but I went through next time. So if we can detail a little bit about each of these advantages that we have, say for example, for diagnosing diseases more precisely, say a genetic test can identify DNA mutations of early childhood leukemia, enabling physicians to choose the correct treatment that fits precisely. Say, for example, for genome-based cancer drugs on survival of childhood leukemia in 1962, we had only a few, maybe less than 5, whereas in 2007, we had more than 80. So the acute lymphoblastic leukemia is the most common form of leukemia, and genetic tests allow us to identify subtypes and allow precise treatment and timing. Today's cure rate exceeds 80%, which was only 4% in 1960s. So this is the advantage that we have. For selecting specific treatment, say for example, a translocation between chromosome 9 and chromosome 22 result in the fusion gene formed from the BCR and ABL1 genes. This fusion on the chromosome 22 is also known as the Philadelphia chromosome and is characterized by chronic myeloid leukemia. This is how it happens. This is chromosome 9 on the left, the longer one, and the chromosome 22 on the right. So the BCR gene is shown in red, whereas the ABL gene is shown in yellow. And as you can see, sometimes a translocation occurs. And if you see on the figure on the right, you will see a fused ABL BCR gene coming on the Philadelphia chromosome 22. So this causes this CML or chronic myelogenous leukemia. And once you have these probes and fluorescence dyes, you can actually see them in the chromosomes. And it results in increased cellular signaling, causing white blood cell proliferation. The imatinib, which is produced by Gleevec, is an oral medication that targets this translocation and arrests this disorder. So this is how this genomic medicine has come. And see, if you can see in the 1930s and all, you had only 2 types of leukemia and lymphoma, a few drugs. And then as we have progressed towards 2020, you have several number of drugs which are in the market and several types of leukemias and 51 types of lymphomas are targeted. So this is the development that has happened over the years. As far as prediction of risk of disease before symptoms appear is concerned, I will give you an example of the BRCA1 and BRCA1 and BRCA2 genes that increase the risk for breast and ovarian cancer. It is a known fact, and it is said that an individual has with a BRCA1 and BRCA2 mutation, they have 50 to 85% more chance of getting a lifetime chance of developing breast cancer than those who are in between without that. So those who are without has got only 30% chance, whereas those with it have got 50 to 85% chance. Similarly, the lifetime risk of developing ovarian cancer with people with mutations in these 2 genes, it is between 10 to 45%, whereas those who do not have the mutation, the chances is only 1.7%. So therefore, there is a lot of risk with those people having this particular mutation in these genes. And there is a hereditary predisposition also for cancers. So these are the predisposition which has been mapped, and it is a autosomal dominant inheritance, high but incomplete penetrance, variable expression, early onset diagnosis, multiple cases of cancer, more than one primary cancer or bilateral disease. So these are the risks that happen, and those of you who follow this, Angelina Jolie also actually, because she had this mutation, she did a mastectomy, and that became a big news all over the world. Now there are both pros and cons, and means there are people who are on either side who generally debate for and against it. But then fact is that this has happened, and that has led people like Angelina Jolie to take actually seriously and take the decision of even a mastectomy. So to that extent, it can actually determine decisions. That is what I want to say here. And molecular diagnosis is at the core of personalized medicine today, and diseases will be diagnosed long before the patient begins to manifest any evidence using traditional tools. So molecular diagnosis come at the center point of the signs and symptoms, in vivo imaging techniques, and in vitro laboratory tests. And managing disease more efficiently, say for example, warfarin is an anticoagulant used to decrease the incidence of blood clots, and it has highly variable response. People have highly variable response to this drug, and there is a tight therapeutic range. And the variants of two genes, CYP2, C9, and VKORC1, account for 30 to 50% of this particular variability. So once we have information on these two genes, and if they are discovered, so then the use of this particular drug will be much more easier. So we can diagnose or predict the risk of the disease, determine whether the treatment is working or will work or not. We can monitor healthy people to detect early signs of the disease, and produce safer drugs by predicting potential adverse events even earlier than before actually initiating treatment, by producing better medical products, by ready access to information, and decreasing healthcare costs. Genetic testing enables new way of thinking about health reforms. So we can actually minimize overutilization, inappropriate care, high costs, and patient safety by impacting the genetic, by using genetic testing. There are examples which I have cited in this particular slide. I'm not going into the detail. Basically, the personalized healthcare actually holds promise, and tailoring of diagnosis, prognosis, and treatment to the individual will improve healthcare outcomes and patient safety. And there are several genes and variants which are now being detected for various kinds of ailments and conditions, and even predispositions. So the more information that we get, the big data that we are generating are actually our repository and the storehouse where actually we can actually mine and then use these into our routine use in practice. Earlier, we used to search for a particular gene. Say, for example, it is like trying to find a lost key in darkness, where we have only one light and search for that. So it has now been replaced with a technique where actually we are having multiple sites and then we are searching. So it is kind of a much better and use of technology to detect whatever we are looking for, as well as those which we are not looking for, but they may actually be helping in our understanding of the disease. So that is how it is being done now. And there are several studies. I am not going into detail, One of the study was the Genome-Wide association study. Another study was the fusion study in Europe. And these actually basically tried to look for genes and their conditions in which various diseases are being affected by mutations in these particular genes. So this is some of the results of the Genome-Wide association studies trying to associate type 2 diabetes with various SNPs or single nucleotide polymorphisms in various genes that occur. Basically, now what we have is that we have a lot of information about various mutations, various genes, various diseases. But the challenge now is that we are developing data in a pace which is extremely high. And we have a huge amount of data available. But to make sense from that, the data is a big challenge now. So the associations of various diseases with particular genes or an interplay of genes, the system biology, the network pharmacology. So these are important areas where we need to concentrate and find out what these information mean. Once the meaning is credibly established, then the industry also can actually look for solutions for these particular diseases. That is what I just wanted to bring to you. These are various depictions of various studies on various cohorts in various diseases. But it doesn't make sense right now because we will not be able to understand them until we study them in context. And in the year 2007, it was said that it was the year of genome-wide association studies because that was the time when these technologies were developing. Now we have a very developed technology, and as I said, that we are drowning in a sea of information, but we don't know how to use that information and how to make sense of it. So the genomic gold rush had started long time back. And we already have this mine which is available with us, but we need to make best use of it. These are all various diseases and genetic loci and when they were discovered that I have listed out in this slide, or rather a number of slides. These are some of the online resources that are available at the National Human Genome Research Institute. You will have several other places where you will get a lot of information on these studies which have been done or are being carried out. These are some of the screenshots of these catalogs of various diseases, the genes responsible, the mutations that happen, even the publications that have been made out of that. So these are all examples, case control cohort studies. This is with interleukin 23R and inflammatory bowel disease, IBD. This is association with rheumatoid arthritis. So these are allocation of various alleles and genotypes. And the various databases are available for this. For example, the genes and disease in NCBI, you can get it at nih.gov. You have various resources, so it's categorized into various disease. So if you click one of them, you will get into them. You have nutritional and metabolic diseases. You have skin and connective tissue disorders. So like that, you can choose your disease and you can see what are the genes responsible and how they interact and how they associate with certain conditions. The OMIM homepage, again, you can find it in NCBI website. You can have the genetic home reference library at jhrnlm nih.gov. So here also you will get information about more than 700 genes and the genetic conditions. So these are various web resources that are available with you. Even Medline also is available on this. You have International HapMap project that also has got a website, and you can go and just have a look at it. And basically, coming back to personalized medicine that I started with, there are various services that have also been started. The clinical genetic testing direct to consumer, where you have online online portals. Nutrigenomics, again, one example. And another one which I liked is 23andMe. So it is an online resource where you can click, and then you will get your kit home delivered. So your kit will come with a this kind of box, and where you will have a spittoon and all. You spit on this, and the sample will be collected, and then you send it back to the courier. You have a prepaid courier which will collect it back from your house, and they will do a variety of tests on your samples, and it is sent back to the company, and then you will log in with your credentials that you have got with your purchase, and you will get your entire report, your clinical reports with your samples, as well as with your predisposition of risks for colorectal cancer, glaucoma, restless leg symptoms. So many are there which you can get your risks assessed with this simple test through your this portal, and you can get clinical reports. You can look at them, and so a wide variety of things are being done, and even your hereditary significance also, you will come to know how your children are also predisposed, or your paternal line. So a host of things you will get. Your ancestry painting also is done with this. So these are some things which we have never thought of, but these are kind of the future. And I will not be surprised if companies like 1mg and other Indian companies, they offer now. Tata has taken it over. So these are kind of things which will be our future. And sitting at home, you can get all this personalized information about your own samples home delivered. So each individual is different, even as a patient. The personalized medicine and treatment is ideal, and a vista of new technologies are now available for further development and research. And each sample that you collect in your routine practice is important, and it can also improve and provide more information on the genetic disposition of the individual towards several diseases. And you can make use of the plethora of the various services that are currently being offered in the line of genomic medicine. And there are several services in India, as I said, they are coming up. And even with our traditional systems also, the Genomics project also is there, which tried to look at the genomic predisposition as far as Ayurveda is concerned. As you all know, it is one system which also viewed individuals differently, not like a single unit. So there is Vata, Pitta, Kapha, and every one of us are guided by different levels of them. And so each individual is different. So again, I go back to our old system. So we are making a full circle and coming back to our old principles of personalized medicine, but this time with the support of a tremendous amount of data that we have generated, and with the use of modern tools. So basically, we are coming back to the future with new technologies, and I am sure that you will be able to make use of this in your routine life, both in terms of research as well as in your teaching duties. Thank you, and I think that completes my presentation today, and I will be more than happy to answer any questions that you may have. Thank you. Over to you.
Thank you so much, sir, for such an amazing session. Now the session is open for discussion. I request all the participants to ask questions directly by unmute your mic, or you can place your question in WhatsApp group.
Hello, good afternoon, sir.
Good afternoon.
Sir, myself Tirupati Rasala, Assistant Professor, Guru Nanak College of Pharmacy, Nagpur. Sir, there is one question. Sir, so much vast research is going in the field of molecular biology and the genomics. Still, there is a concern of the life-threatening disease and disorders.
Right.
So is the molecular biology is going towards treating the life-threatening diseases or disorders? At what time we are going to achieve our target? So can you elaborate on this, sir?
Yeah. The beauty of it is that I'll tell you from my own experience, the more I learn, the more I feel that I have come to nothing. So I think the same thing applies for our knowledge globally. As I was talking about systems biology, is that you introduce one drug and the entire metabolism shifts to certain direction. So these are new information that we are generating. So if you look at the pharmaceutical market now, mostly in developed economies, more and more emphasis is now given to a kind of what I will call, what is the term I'm forgetting, but not the new chemical entities, but it's more of mixtures together. So more of trying to address the root cause rather than the signs and symptoms. So like now number of diseases also have increased, the conditions have increased. As I said earlier in one of my slides, the lifestyle changes that we have, the climate change that is happening, so these are all causing a plethora of new conditions put into us, as well as you have the old pathogens which are emerging with mutations, and you have drug resistance everywhere. So challenge is actually much more than the solutions that we have come up with. And as I said, that we have a lot of hope in this molecular tools and molecular medicines and all, whichever are coming. But simply the making of sense and then coming up with concrete solutions for particular diseases has not been able to keep pace with the new diseases that are being developed or the re-emergence of old diseases. So that is the challenge that we foresee. Any time frame, I don't know how we are going to come up, but I am sure this COVID pandemic has shown us how underprepared we are, and such kind of infections are going to come, and we have to deal with it.
Okay, sir. Fine. Thank you, sir.
Any other question?
Good afternoon, sir. Very interesting presentation. And this is Dr. Nitya from American College. I have one question that we have many more molecular tools for analyzing nutrigenomics or pharmacogenomics, and now it's evolving nutritional epigenetics. So will you say some tools can be used to identify or the diseases and its interaction with relevant to lifestyle changes, that is we call it as epigenetics. Rather though we don't have any mutation or genetic variations, what is the role of epigenetics? Kindly, sir.
Yeah, I would personally say that epigenetics has got a much major role than it is anticipated. We do talk about predisposition or your risk factors, which are genetic traits or characters.
Yes, sir.
But you look at what happened with Dolly, the sheep. So it had premature aging, isn't it? It had premature aging, and ultimately it died early.
Yes,
and so this is one area which actually not much of research is sometimes legally allowed, and not much has been done also. But I think epigenetics plays much bigger role, if not equal, to the genetics of it. Because what I feel is that, see, we know only a very small fraction of our genome. The remaining fraction we don't know. So is it possible, say, for example, if, say, I am predisposed to, say, certain types of cancer, but there may be quite a number of other genes which are in what we call as junk DNA that may be actually helping me prevent that. It knows, my system knows that I am predisposed, so it is actually having a role in kind of suppressing it. That is also quite possible, which we have not discovered. So basically epigenetics also is very important, is that even if I'm predisposed, then what are the other factors which we do not know about, other than the ones which are discovered? So, they have a very important role, and those are things also which need to be taken into consideration. One of the examples I will tell you is that this now a lot of work is being done on this metabolome, particularly gut microflora. So they are associating almost everything with certain types of microbiome. You do something on microbiome, it gets published, and you will find some association with certain things. But do they really make any difference? I don't know. So even in Ayurveda, there is the panchakarma that they do. One of the procedure is to actually have your entire gut cleaned. They will be feeding you with only all jari buti and all herbs and all, and then they will be feeding you with only ghee and something, and then basically they will be washing away all your so-called toxic flora what you have, and then a new kind of flora will be implanted. So why we take so much of curd and this thing after we take antibiotics or after we recover from a disease? It is to replenish with some good microflora in your gut.
Yes, sir. Yes, sir.
So my microflora today may not be the same after I do some panchakarma or something. So similarly, it can change with various conditions. So these are things which needs to be looked into, and we do not have enough evidence for this to draw logical conclusions. So these are things which actually are very important, which maybe you and I need to see and do some kind of a credible research to find out.
Yes, sir. Sure. Thank you, sir. Thank you so much.
Thank you so much, sir. Now I would like to request Miss Jidnasa ma'am.
Thank you. I would like to thank Dr. Roy sir for such an informative session. During the session, sir discussed about evidence-based medicine. Further, sir discussed principle, different forms, limitations, and applications of polymerase chain reaction. Sir highlighted DNA and protein sequencing and progress in genotype technology. Also, sir focused on principle and applications of microarray, genetic variation in complex diseases, advantages of genetic tests, application of molecular diagnostics in personalized medicine, and benefits of personalized medicine. On behalf of Bharati Vidyapeeth College, I express my sincere thanks to you, sir, for sharing your valuable knowledge with us. Your magnificent speech made session so delightful. Thank you once again, sir.
Thank you all of you, and thank you once again to all the organizers. Thank you.
Thank you, sir. Now I request all the participants to turn on your camera so that we can have a photo with our eminent speaker. Thank you, everyone.
Hello, sir. Hello. Roy, sir. Hello. Hello.
Dear participants, we are starting with our valedictory program. Please be connected. Good afternoon, all. I, Ms. Asha Jadhav, on behalf of Bharati Vidyapeeth College of Pharmacy, Kolhapur, would like to welcome you all for the valedictory program of AICT-sponsored Faculty Development Program on Nutrigenomics Unveiled Frontier in Healthcare, organized by Bharati Vidyapeeth College of Pharmacy, Kolhapur. It gives me an immense pleasure to welcome our guest for valedictory function, Dr. Subranna Roy, sir. We really feel honored to have you, sir, with us today. Now, I would like to request all the participants to share their views regarding this faculty development program. Dear participants, please share your views regarding this faculty development program.
Hello, ma'am.
Hello.
Yes, ma'am, continue.
This is Dr. Nitya from American College. Shall I proceed?
Yes, ma'am, please.
So, first of all, I thank the ATAL Academy for organizing such a wonderful faculty development program on Nutrigenomics. So, it is a needed one, and the sessions, all the sessions are very interesting and thought-provoking. And I must congratulate all the members of this team, that is the organizing team, for giving such an eminent speakers for this session. Thank you so much.
Thank you so much, ma'am. Anyone else from the participants, please share your views.
Hello. Good afternoon, madam. Am I audible?
Yes, sir, you are audible.
There is some disturbance. Am I audible, madam?
Yes, sir, you are audible, sir.
Okay. Thank you, madam. Thank you. Good afternoon, one and all present here. Myself, Tirupati Rasala, Assistant Professor, Guru Nanak College of Pharmacy, Nagpur. First of all, I would like to express my gratitude towards the whole organizers of Bharati Vidyapit College of Pharmacy, Kolhapur. The coordinator, Dr. Tambole sir, for giving this opportunity to attend this 5 days faculty development program, which was organized by, which was sponsored by rather AICTE. So, sir, it is one of the best, well-organized program which I have attended. The speakers were highly committable and having eminent personality in their respective fields. I am very much impressed with all the speakers, starting from on first day, Dr. Abhay Pirsulkar sir. Then there was Dr. Ladda sir, Srinivasan sir was there, Suraj Ratnakumar sir was there. I think Gilda sir was there on entrepreneurship program. He has given the excellent thought process. Then Annapuri sir was there, Itankar sir, Rao sir, then right now the Subhana Roy sir. So all the speakers were very well said, and so this is a kind of program that we got excellent knowledge. Rather, what you know. So in addition to that, we got additional knowledge. So for that, we oblige to all, sir. It was fantastic conference, no doubt about it. And effective organization. I'm always impressed with such kind of commitments and efficiency of those who plan this kind of organizations. A very good organization, sir, no doubt about this. And I really enjoyed and having a wonderful experience and looking forward to attend such kind of program in the future also, sir. Thank you very much. Thank you, coordinators. Thank you all the faculty and each and everyone and all the participants. Thank you very much.
Thank you so much, sir.
Thank you,
Anyone else? Anyone else from the participants, please share your views regarding the faculty development program. Moving forward, now I would like to request our eminent speaker, Dr. Subranna Roy sir, to deliver your valedictory address. Over to you, sir.
Thank you. Once again, I would like to congratulate the organizers for such a wonderful faculty development program that had been stretched over this 1 week, and for inviting me to speak on this particular occasion on this valedictory program. It is very heartening to hear from the faculty, the participants, that they have been benefited from this program, and the quality of the speakers. And yes, we are going through tough times. We always like to be physically present and interact with each other. But in these times, there is no other way other than this hybrid or this online mode. And it is very challenging to organize such programs purely online. And I give due credence to the organizers for doing it so wonderfully well. And I had also attended, as I said, a couple of years ago, this program. And I have personally seen how much efforts have been made to actually deliver the goods of this faculty development programs to the participants. And we are going through tough times actually, because we have been resetting our lives both at the professional as well as personal levels because of this pandemic. And perhaps it has given us a lot of lessons, and which we have learned in this pandemic. We have also got the opportunity to reflect on many of the things which were perhaps not needed And with this backdrop, we have also learned many things. Say, for example, I myself, I think I completed some 20 online courses during this 1 year, when sometimes I was unable to go to my lab or unable to do some work. And that has provided me an opportunity to do these courses, which otherwise during routine times I would never have got opportunity or time to do that. And our learning never ends. So till the last day that we live is a learning experience for us. And it is our mentors, our professors, our teachers who actually play a major role in our own development. And since you are all teachers yourself, so naturally you are privileged to change the lives of so many number of people. And I myself don't forget my teachers, my professors, and naturally your students will also never forget you. So the more you take uh, lessons from these small events and even the larger events that come to our lives, and then try to use them practically in sharing our experiences with our students. So I think that is what we are meant for. And we make our students much more practical oriented and much more rooted to the ground than otherwise they would be only by learning theoretical aspects. And with this, without taking much of time, I would like to conclude. I have already spoken almost about an hour on whatever little I know. It is something related to the personalized medicine or the genomics part of it. And as I said, the key that the branches and sub branches are increasing so much. So nutrigenomics is again another thing which was not in that list of the busy slide that I showed with the list of the disciplines and sub-disciplines that we have now. But yes, it is another one that is coming up, and again, which is extremely important. Our nutritional aspects have actually gone from bad to worse, and these are some areas which we need to concentrate, particularly in research, and then bring evidence to actual practice. So with this, I would request all participants to take good memories of this particular learning experience and try to use them to the best in your routine professional careers. And I wish you all the best. And once again, I thank the organizers for giving this opportunity to speaking to you. Thank you. Thank you very much.
Thank you so much, sir, for your valuable address. Now, I would like to request coordinator of the program, Dr. Firoz Tamboli, sir, to brief the program and propose the vote of thanks.
Thank you, Asha ma'am. Chief guest of today's function, Dr. Subarna Roy, sir, our beloved principal, Dr. H.N. More, sir, Vice Principal, Dr. Bhatia, sir, dear delegates, and my dear colleagues. I am here to brief you about what we have gained in 1 week as it is sponsor ATAL FDP on Nutrigenomics Unveiled, Frontier in Healthcare, which was successfully conveyed between 23rd November to 27th November. I am sure you might have been benefited a lot with understanding of various nutrigenomics for non-communicable disease, nutrigenomics ethics and challenge, precision diet and nutrition with genetics, nutrigenomics a customized challenge for pharma industry, nutrigenomics present and future in healthcare, and in silico tools in nutrigenomics and molecular tools in pharmacogenomics, etc. Understanding of these things may help you in your journey towards an effective faculty to teach nutrigenomics approach and as a researcher too. You might have also gained an adequate insight into future developments which are likely to take place in the preparation of nutrigenomics, nutraceuticals, its importance and relevance in the human life. I believe the objective of this FDP have been met. At the outset, we would like to thank AST for sponsoring this FDP to us. Without funds, program would have hardly impossible. Secondly, I am extremely thankful to our beloved principal, Dr. H.N. Morris sir, for his continuous support and cooperation. Next, I would like to thank you, our guest for inaugural and validatory function, Dr. Abhay Harshurkar sir, Senior Scientist, Irsha Pune, and Dr. Subarna Roy sir, Director, ICMR, NITL. I would like to thank all research persons for their valuable time. To name few, Dr. K.S. Ladda, Dr. Srinivas Gope, then Dr. Suraj Ratnakumar, Founder Scientist Swajan Chennai, Dr. Suyit Gilda, Professor Uday Annapure, Director ICT Mumbai, Dr. Prakash Itelkar, Dr. Sachin Jagtap, Dr. Ajay Namdev, Dr. Ajay Agwane, Principal Scientist NCL, Dr. Amol Raut, Dr. Praful Chaudhary, then Dr. Surajit Devna. I am extremely thankful to all speakers for their valuable time sparing. We had totally 14 deliberation in area of nutrigenomics and almost covering all areas. I express my deep gratitude towards them. We are very thankful to all delegates, those who have online joined from different places all over India. Especially thanks to Mr. Tirupati and Dr. Nitya for his active participation. I am thankful to all principals for deputing their faculties. At last but not least, I am thankful to my college teaching faculty, non-teaching staff, all committee members, coordinator Vishal Thora sir, Professor Rohan Kumar Sawan, Professor Mrs. Asha Jadhav ma'am, and Anga Azrekar, without whom this program would have been simply impossible. At the conclusion of program, I firmly believe that the program had a good take-home value and has benefited you all. I wish you a very best to come in your life. Thank you.
Sir. Thank you so much. So here I would like to announce we are concluded with this AICT sponsored faculty development program on Nutrigenomics Unveiled Frontier in Healthcare organized by Bharati Vidyapeeth College of Pharmacy, Kolhapur. Thank you so much all for your cooperation and support. Without you, we cannot achieve the success for this program. And here I would like to announce the link for the attendance and the link of exam will be shared on our WhatsApp group. Thank you all. Thank you so much, sir. Thank you so much, Roy.
Okay, sir. Thank, thank you very much, sir, once again.
Thank you, sir. Thank you.
Hello. Hello. Hello, ma'am. Hello.